Hi everyone, I'm Dave Robertson from Vilsera. Today I'm excited to introduce the new External Connections panel on Biodata Catalyst powered by 7bridges. This feature significantly improves how you manage your connections to various data ecosystems, making your research workflow more efficient. We'll cover how to monitor connection status, manage your connections, and import data across platforms, all from a single, intuitive interface. So here's what we're going to cover in the next few minutes. I'll walk you through this new feature, show you where to find it on your dashboard, and then we'll explore the three main platforms you can connect to. Cavatica for pediatric research, Synapse Portal for collaborative work, and Cancer Genomics Cloud for datasets from the Cancer Research Data Commons. We'll also chat about how to get access to controlled datasets, because let's face it, that can be confusing sometimes. And to wrap things up, I'll show you a real-life example of pulling data from CGC straight into your Biodata Catalyst project. It's actually pretty cool how seamless it is. Let's start with where to find this new feature. The External Connections panel now appears directly on your Biodata Catalyst dashboard in the bottom right corner. This gives you immediate visibility into your connection status with external platforms. Notice the green check marks indicating active connections and gray text showing inactive ones. This immediate feedback helps you quickly identify if you're ready to import data or if you need to reconnect first. We've designed this to be as intuitive as possible for you. Here's a video of me navigating the Biodata Catalyst powered by 7bridges platform dashboard. You can see I have a bunch of projects, but when I scroll down, you will see the new UI on the bottom right corner. I zoom in for you to get a better look.
Let me demo for you the Manage Connections window. So you will see down here the link to get to the Manage Connections screen. So we'll see the three connections here, and I'll get into what those are in a moment. But what you want to take note of is you have an expiration date, which is generally a month from whenever you last officially logged in. But it could be less. So you always need to, if you're troubleshooting a connection, always come here and check it out. So you'll see I'm not connected to Synapse, but I am connected to Cavatica and Cancer Genomics Cloud powered by 7bridges. So let's just demonstrate how it would be to disconnect. So let's say you want to deactivate. You would simply click deactivate, and then you could go ahead and deactivate this one too. And then let's connect this one. So when you connect, it's actually going to send you to the other platform and have you authenticate. And to learn, there's a link to learn more about each of the individual platforms. So I'll just log in. Authorize. Let me go back to the original platform, Biodata Catalyst. You'll see that I'm now connected. That's a quick way to check using the External Connections panel. And then we can see that I'm connected here. So it's just as easy as that. Those two windows provide a lot of utility.
Our first integrated ecosystem is Cavatica, specializing in pediatric and rare disease research. It hosts comprehensive datasets from several major initiatives. The NIH Common Fund's Gabriella Miller Kids First Pediatric Research Program houses data from over 30,000 participants across numerous childhood conditions, including structural birth defects and pediatric cancers. Include investigation of co-occurring conditions across the lifespan to understand Down syndrome, offers unique genomic datasets on Down syndrome and associated conditions. Cavatica also provides access to the Childhood Cancer Data Initiative, CCDI, and specialized datasets from Children's Brain Tumor Network, CBTN. When you connect Biodata Catalyst to Cavatica Through the External Connections panel, you gain direct access to all of these specialized pediatric and rare disease datasets without downloading and re-uploading files. This integration is particularly valuable for researchers studying developmental disorders, childhood cancers, congenital anomalies, or rare diseases. The connection uses the same standardized DRS protocol as our other integrations, ensuring secure and consistent access across platforms while maintaining appropriate data access controls. The second connection is to Synapse Portal, which serves as a collaborative research network across multiple disciplines. Synapse hosts datasets from initiatives like AMP-AD for Alzheimer's research and PsychEncode for psychiatric genomics. What makes Synapse particularly valuable is its community-driven approach, where researchers can not only access data, but also contribute to growing knowledge bases. When connected through the External Connections panel, you can directly import Synapse datasets into your Biodata Catalyst workspace, facilitating cross-platform analyses that might otherwise require complex data transfers and duplicated storage. Our third integrated platform is the Cancer Genomics Cloud, or CGC, which houses comprehensive cancer research datasets. CGC provides access to the Cancer Genome Atlas TCGA, target pediatric cancer data, and other critical oncology resources. The platform specializes in supporting computationally intensive analyses of large-scale cancer genomics data. By connecting CGC through our External Connections panel, you can seamlessly import these massive cancer datasets directly into your Biodata Catalyst projects without storage duplication. This integration is particularly powerful for researchers working across both cancer and non-cancer contexts who need to leverage diverse dataset types. Before you can connect to many datasets, you'll need appropriate access permissions. Start by checking your current access using the Check My Access button in Biodata Catalyst. This shows which datasets you've already authorized to use. For additional access, you'll need to submit requests through dbGaP, providing your research purpose and institutional affiliation. Once approved, those permissions will be reflected in your Biodata Catalyst account, typically within 24 to 48 hours. Remember, the different data sets have different requirements, so it's worth checking the specific criteria for the data you need. The External Connections panel makes it clear which platforms you can access, but the underlying dataset permissions are managed through dbGaP process. Synapse and CGC have some open data sets that will be accessible right away. Now let's explore one of the technologies that powers our external connections, the Data Repository Service, or DRS protocol. DRS is a GA4GH standard that provides a universal way to access data across different repositories. Think of it as a universal translator that allows Biodata Catalyst to communicate seamlessly with connected platforms like Gen3, Cavatica, and Cancer Genomics Cloud. What makes DRS revolutionary is that it allows you to work directly with data without downloading entire files. When you import data this way, you're actually importing DRS URIs, unique identifiers that point to the data. The platform then fetches only the data you need for your specific analysis as you need it. When you see a file imported via DRS in your project, you'll notice a special indicator showing it's an external reference. You can use these files in your workflows just like any other file, but they don't cost anything for storage. This makes your analysis both faster and more efficient, particularly valuable when working with large genomic datasets.
Okay, now I'd like to show you what it's like to use the external connections feature and import from the Cancer Genomics Cloud. So I have a new project for us called External Connections Demo, and I made some notes here. So we're importing from CGC. What we need to do is first check the External Connections panel and make sure that the connection is live. We need to go to the CGC and prep the files, which I've already done, but I'll show you what it looks like. We need to export DRS manifest. We need to come back to this project and open a Files tab in this project and select Import from DRS manifest. So roughly 4 steps. Okay, so first things first, let's check the CGC connection. So if I go back to our platform dashboard and then quickly see that it's connected. So that's good. So I'm going to go back to here to our project. And so the next step is to prep the files on the CGC. So what this involved was signing on to the CGC, also known as Cancer Genomics Cloud. And I went to the data browser and made this data browser flow. If you don't know, the data browser is also currently available on Biodata Catalyst. It allows you to make a visual filtering flow of and selecting the files that you want. So in this case, I'm picking disease type glioma, only the primary tumor. I'm choosing open access, and I want the RNA transcriptome profiling files. Now, if I did have controlled access, I could, of course, do that as well, but I would need to make sure like my dbGaP authorization was in place to be able to move those or to be able to link those to another platform. So it's showing 671 cases, 672 samples, and 1,697 files. And you can see all the files here. The green check box is, the green unlocked button means that it's open access. So the next thing I do is I copy the files to the project, which I've already done, but the button's up here. Then I go to the next step here is to go to the Files tab in the Export project. And you'll see that it's indicating that this is a DRS link. So I'm just going to grab the first page of them, or the first what's showing here. If we wanted to, we could grab all of them. That wouldn't be a problem. But I'm grabbing the first 14, it looks like. And I'm going to generate a DRS manifest. Okay, and then it's going to download. Or actually, it's going to open in the project, and I'm going to download it. And I've already done that. We've done everything we need in a CGC. Now we're going to go back to the Biodata Catalyst. And it says the next step is to the Open Files tab in its project and use the Import DRS Manifest option. So I'm going to go to Files. I'm going to click Add Files. And it has all these options. What I want to use is the GA4GH Data Repository Service, or DRS, as it's commonly known. So I click that. Now I can paste them directly in here, but I can also import from a manifest, and I'm going to upload from my computer. In the DRS, my DRS folder where I downloaded it, the demo folder, I'm going to click Open, and I see it here. And then you can add additional tags if you want. You could either to auto rename or skip. I like to do auto rename just in case there is a naming collision. It'll make sure that there's a prefix file name so it won't right over top of it. And click Submit. And then what you'll see is first it imported the DRS manifest for reproducibility reasons. And then if you click on the bell icon, you'll see that it's already imported all 14 of them. And I can check all the details here. And you'll see that these are some of my other imports I've done. And you'll see that it was able to successfully import all of these files using the GA4GH API. So if I go back, it should refresh, and then you'll see that it's been refreshed. These are all of the links, and this icon here means that it's a link and not a file. So remember, we're actually just linking back to the CRDC's datasets, and this is not actually duplicating any data, but I still get all this information about these particular files, including lots of metadata. So we have all the information in the case, the disease type, the primary site, and everything that you might want.
The External Connections panel is available now to all Biodata Catalyst users. There's no additional setup required. Simply log in, and you'll find it on your dashboard, ready to connect you to these valuable data ecosystems. For comprehensive documentation, including step-by-step tutorials and troubleshooting guides, visit the Biodata Catalyst website. Our support team is available through the Help button in the platform interface if you encounter any issues. Thank you for your attention, and we hope this new feature helps accelerate your important research work.